Good morning and good afternoon, America. My name is Marina. I'm the product manager here from Singer Instruments, and it is my pleasure to welcome you from our humble studio here in Minehead for our webinar on automating PCR preparation. And we hope to answer the question of, is it worth it? As you may or may not know, Singer Instruments has been in the lab automation scene for over 40 years. Initially, we started with yeast genetics, but now our customer base encompasses a diverse disciplines, model and non-model organisms that you can name in over 60 countries. This is why I'm so excited that we are branching into a new technology for us, automated liquid handling. So I wanted to introduce you to SQWERTY, our low to medium throughput pipetting robot, your everyday pipetting sidekick, ideal for any lab, for automating those manual, repetitive, and frankly boring pipetting jobs you have to do on the daily. So the webinar's focus will be on my experience in a couple of months of getting to know SQWERTY and transferring my manual PCR protocol on it, what I have learned, and what are some of the considerations I had to take into account when trying to automate my workflow. Now, I'm new to automated liquid handling. SQWERTY is the first automated liquid handler I've used, and I have no prior knowledge or skills or coding or anything like that. So I'm hoping that my experience will paint you a clear picture of what you can expect. So before I just set up SQWERTY to run the workflow and kind of explain to you how I approach programming it, I wanted to give you a broader overview of the product. So as I said, it's our benchtop pipetting robot. It's got a manual door and it's got 3 deck spaces. Now, the leftmost deck is usually reserved for tips. All of our tips are compatible with a TCAN cone, so it's not Singer proprietary automation tips that you need to be getting, whereas your other deck spaces are available for your source racks or plates and your destination racks and plates. Today I will be using our 1.5 to 2 mil tube holders, but I also wanted to show you another accessory that you can fit reservoirs into. This way, if you've got a larger volume that you need to be transferring out of a pool, instead of stacking up a couple of tubes with in the rack, you can use a reservoir instead. For the destination plate, I have chosen a very bog standard PCR plate, skirted and SBS format. That is the format compatible with SQWERTY and all of our decks. You can customize pretty much any standard labware as long as it conforms to the SBS format. You can program it into the user interface, or you can even experiment and like my colleague Fiona, who has recently adapted one of the bays to hold a 3D printed version plate adapter holder for a multi-top target plate. So yeah, pretty much you can customize it in any way you want. But let me set up the deck spaces for today. As I said, just some tube holders. They fit into the decks nicely. A1 always in the bottom left corner, and you can use this handy screw to secure your plates in place. That way they are certain not to go anywhere. SQWERTY offers a broad range of volumes separated across 3 pipette modules. Now, the default pipette module that comes with SQWERTY is the 250 microliter one. Offers quite a nice broad range of accuracy down to 5 microliters. For the purposes of my application, which is colony PCR, I'm transferring a very low amount of my template, only 1 microliter. This is why I decided to use the 50 microliter pipette module, because it will give me the best accuracy in that low volume range. But obviously, if you need a broader range and you're working with bigger assays and reagents, you can purchase the 1000 microliter pipette module as well. Which pipette module you're using will depend very much on your protocol, and it might surprise you that not all protocols or all steps in certain protocols require the same degree of accuracy. This is why I have chosen to couple the 50 microliter pipette module with 20 microliter tips, and I am using sometimes multiple transfers because of that tip capacity, because the most important thing for me is to accurately transfer the low volumes of my DNA template. So before we get into the nitty-gritty of the workflow programming, I wanted to share what is my favorite feature of SQWERTY, and that's the interactive mode. Now, when you think about automated liquid handling, many people ask, why is it so complicated? Well, if you think about a manual pipette, it's really dependent on visual cues. You adjust your pipetting parameters based on the liquid that you're pipetting, your receptacle, but a robot just isn't able to be as nuanced and rational as a skilled pipetting pro like myself. At least not yet, but we're working on it. This is why we have introduced something called liquid classes. You can think of liquid classes as being a set of pipetting instructions designed specifically for that particular liquid. And it's really the most important way that you're going to be reaching the level of accuracy in pipetting with any robotic platform, because you need to be able to program those adjustments that you usually take for granted because you do them visually and manually. For example, if you're pipetting glycerol, you know to aspirate more slowly and to wait a little bit for all the fluid to go up the tip, otherwise you'll be aspirating air. If you're submerging it to low, you're going to cover the outside of your pipette tip with gloop, so accuracy goes out of the window. On the other hand, if you've got volatile liquids like certain alcohols, you're going to notice significant drippage, and those are all the things that we can control and manage with SQWERTY. So let me show you what the interactive mode looks like. Why is it called interactive? It basically means that you're not have to program an entire workflow and repeat it and replay it over and over again to make sure that your pipetting parameters are right. The interactive mode is there to give you an opportunity to focus on the simple transfers. You don't even have to select a destination plate like I did here on the right. You can have a tube in A1 that you want to be pipetting into tube located in C1, just to make sure that whatever reagent you're trying to optimize is getting pipetted properly. So what can you actually do? So it's asking me to select the source item. Now I'm not going to be showing you this live because of health and safety issues. We have chosen to work with food coloring and pre-prepared reagents, but I'm hoping to show you in the software how visually it works and how intuitive it is actually to get it do what you want it to do. So I've selected my source item. Let's pretend that there's some heavy glycerol in there, and I can now set how much I want to be aspirating. Now, because I've let SQWERTY know the deck layout positions, so exactly what is happening and what is located in each of the bays, because again, our SQWERTY doesn't have eyes yet, it has limited the aspirating, so the taking up of the volume, to the tip capacity, which is 20 microliters. If I want to be pipetting 10 microliters of glycerol, I can either use the plus and minus icons, or I can simply put in the numerical amount, and you'll see that SQWERTY immediately visually shows you how much of the pipette tip will be taken the liquid up into. The same way is later on in your workflows, you'll be able to see exactly how your liquid levels are getting tracked in order for you to set an insertion depth. So that's the parameter right next to the volume range, and currently it's set to go into the bottom of the well, but you can again use either the minus or plus buttons, or simply put in at what level you'd like the tip to be immersed into. Now you might think that's too much of trial and error, and depending on whether you know how much liquid is in your sample, that may be true. But this is why we've got liquid level detection. So it's this setting right here. Liquid level detection is basically pressure-based sensing of the surface of the liquid in the tube. So what you're actually doing with enabling this setting is, instead of letting it know how much you've got in the tube and telling it where to start aspirating from, SQWERTY will start going into the tube until it senses that pressure threshold you've set up, for example, for water, glycerol, ethanol, broth, and it will detect when it has reached the surface and start aspirating right there. This can be coupled easily with liquid level tracking, so something not really useful in the interactive mode because we don't have a set of operations, but it will track the liquid levels in the tube over time. To really increase the power, the accuracy of aspirating and dispensing, especially for those viscous and volatile liquids, for the viscous ones, we've got the blowout. That's your equivalent of a second stop on a manual pipette. And it just basically means that you're getting rid of all of the residue within the tip. You can set up in the advanced parameters how big of a volume you want that blowout to be, but it does have a tip capacity, so you can't be putting up a 20 microliter blowout here because I'm already taking up 10 microliters of the tip's capacity, so my blowout is limited to 10 as well. Apart from just the volume of the blowout you'd like, especially for glycerol I had to crank it up to 15 I believe, you can also fiddle with how fast that blowout is being made. So for some really sticky liquids you can increase it and see how that affects your accuracy of transfer. On the other hand, you can enable the travel air gap, which is really handy for volatile liquids. Now, if you've got a drippy liquid, when you're aspirating from a tube, even manually, but also on liquid handlers, by the time you get to your destination tube, you might already get significant dripping. Usually ethanol is the culprit. What the travel air gap does is it takes up, after you have aspirated your ethanol, for example, takes up a little bit of air to give it that buffer and prevent the drippage while the pipette module is traveling to its destination. Again, you can customize how quick that happens, the flow rate, and what the volume of that travel air gap is. Again, be careful because it does add to your tip's capacity. I am happy with my settings for aspiration, so that's when you go and configure your dispense settings. Now I'll pretend like I've got a destination tube in C1. SQWERTY has again visually let me know that the destination tube is colored in red. And here, similarly to the aspiration step, you can set up how much you want to be dispensing. Now you can use reverse pipetting and dispense less than you aspirated. That's good also for some viscous liquids if you want to be making sure that you're accurately transferring. A second really useful thing here is setting the immersion depth, because I like to go close to the bottom of the tube depending on really the reagent, and you can control your immersion in that manner. What you can also do in the dispensing step is you can enable a mix, enable and customize a mix. So the mixing works not in a way that the pipette module will start revoluting or circling the sample. It actually means that it's going to take up a certain amount. Usually I put it the maximum of the tip's capacity, so 20 microliters. You can control how fast it's going to be aspirating the mixing to prevent any bubbling for bubbly liquids, and set up how many cycles of aspirate and dispense with that mix volume you want it to make. You can also set the mixing insertion depth in case you don't want to be going too far, or making sure that you're not aspirating air. But don't fret, because we got mixed liquid level tracking as well. So usually it'll find the surface of the reagents very well. Now, as you've started to experiment here in this interactive mode, you can press play after each setup and just keep a close eye on how you've programmed that actual step. Is everything getting dispensed from the tip? Are you happy with the amount that it's being pulled up? Does it look like there's some leftover residue? And you can fiddle with the settings until you're happy. Why does that matter? We can then save those settings into the liquid class. Now you can open the liquid class editor by pressing on the settings button there. You can see on my left side that there's already some pre-made ones I've made in preparation for today. There's even more advanced settings, whether you want to be dwelling after you have aspirated. So for example, to get that last bit of glucose in, and all of the flow out rates. The important bit here to enable successful liquid level detection is to make sure that you experiment with the pressure threshold. So if you are trying to optimize a certain reagent and you're seeing that it's not getting adequately sensed, you can increase or decrease this pressure threshold and the speed at which SQWERTY is being submerged into the tube to get the parameters that you need. After you have done that and are happy with the settings that you've made, you can close it down, and not right now because I haven't changed anything. SQWERTY will ask you if you want to be applying all those parameters to all of the workflows in there. So you can retroactively add in those settings to your workflows as well. I think that's about everything I can cover in the interactive mode. So let's go into the workflows. So the point of saving the liquid class is that you basically only have to do it once per reagent. You don't have to every time try and figure out what settings you applied and change them manually. But if you are setting up a brand new protocol, you've got a couple of reagents, it will take you a little bit of time just to get those pipetting parameters right. So let's get into the actual workflow. We've got some pre-made ones here, and I would like you to have a look of the layout of the user interface before I set it to play. Because once it plays, we can't go back into the user interface, so we are not messing with those liquid parameter settings. So the way that you can build workflows in SQWERTY is by the use of operations, and those operations can be as granular or as grouped as you would like, but it always makes sense for troubleshooting and for changes to workflows to group similar tasks together. So what do I mean by that? I could have, for example, made two separate operations for the addition of the forward and the reverse primer, but I thought that would be inefficient. So what I did was I grouped them together, made one step to cover both of them, and that way, if I'm later on want to make some changes, I'll not wonder if I skipped to apply changes to one and not the other. you can program a couple of different types of operations into SQWERTY. I'm going to be walking you through them as we get along, but they do increase in the level of easy or complexity depending on what you need from that particular workflow. Now you can, I wanted to show you, I'm going into the actual operations to show you that you can apply the liquid classes per the operation step, and as well you can, for the certain steps, you can adjust the blowout volumes, air gaps, and how far the liquid level detection will be traveling. I won't build a new operation right now, but I wanted to give you a little flavor of what you can do. So if I wanted to add another step to my workflow, we've got guided transfers and guided multi-dispensing, so like aliquoting. Why it's guided, it's the best feature of all, I think, because you don't have to do any of the hard work. All you need to do is, in a guided transfer, set your source, pool, tube, whatever, set your destination tube, and specify to SQWERTY how much you want there to be in that final tube. SQWERTY will then automatically calculate what types of transfers it's going to be doing, how many it's going to do based on the tip capacity and the pipette module for that perfect accuracy you want to be achieving. If you want a little bit more complexity and control all of this yourself, you've got advanced transfers, advanced multi-dispense, and we have prepared a serial dilution operation as well. Another favorite of mine is the UV function. I love to run it before and after my workflows, but SQWERTY is already really easy to clean anyways. So let's start with running the workflow. So I'll press play, and once I press play, it will just ask me to make sure that all of the decks have got the objects that we have, that it's expecting. Now, as you can see, I was testing the workflow earlier and I didn't have a lot of tips, but I've put a fresh one in, so I don't want SQWERTY to start grabbing tips from this far down in there. It's really easily replaceable by just clicking onto the next available tip. Even if you've got people who are chaotic and skip the tips that they're taking on, don't fret, because SQWERTY has got automatic tip detection, so if it finds an empty spot, it will just move along and find the next available tip. And hopefully I have demonstrated to you how easy it is to troubleshoot this. No need to call an external engineer or do any complicated programming, but I'm hoping from now on it will be smooth sailing. Okay, so yeah, for the addition of the water to the PCR master mix, I didn't have to be fiddling around with the pipetting parameters, so I've used the default water liquid class. So the software allowed me to set the initial reagent volumes. This way I was able to kind of optimize the liquid level detection in order to take the shortest amount of time. Now you can help the pressure-based sensing of the surface find the surface quicker by eyeing up how far along the tube you think it's gotten to and setting the insertion depth there to allow it to start that liquid level detection protocol and move on. And I believe I've already mentioned, but in case I didn't, the traceability of naming the individual source and destination tubes and wells really comes in handy when it comes down to reporting. Both in the report, which is exportable in PDF and CSV formats, you can be looking at the console, which kind of breaks down even further what operation SQWERTY is actually doing. This is really good for the troubleshooting log, and I'll definitely be consulting it after this webinar. Just so I don't have to be migrating from the software anymore, I'm going to be showing you what the report looks like here. It's pretty much the same layout as you can see on the right side, except of being primarily white and black. So hopefully with the GoPro you were able to see that SQWERTY is currently adding in the primers, the forward and the reverse, and if you remember, I have programmed in a mixing cycle after these two in hopes of homogenizing the master mix that we've been preparing so far. Now, as I said, you could do this after each one, but I was saving on time, and visually I could observe that the quality of the mixing was sufficient enough to only program it into that step. After the primers have been added, this is kind of the last time that we will be the last thing that we will be adding into the master mix. We have been going on to transferring the actual colonies, the DNA template. Now, the good news is that resuspended colonies in molecular grade water also don't need their separate liquid class, so I was able to apply the water one and it transfers it beautifully. I was concerned because I was transferring only 1 microliter, and I'm hoping that with the yellow liquid you're going to be able to visualize this as well. Again, in the way that I've programmed it, I could have added the master mix first into the destination plate. To keep the webinar time friendly, I have only set up 9 reactions, so 3 biological replicates and 3 technical replicates per colony. And I've decided to first add the colonies into the destination plate, and hoping that adding a larger amount of the master mix will already help homogenize the template with the master mix in the later step. Despite that, I have programmed in a mix, so after the master mix is getting put in, we're going to be into each well, it's going to be mixing it with the template as well. So with the colonies, I have also would like to note that I've decided to reuse the tip because I've noticed the volume accuracy when I was measuring it was good. And again, I just like to save on tips, save on reagents, and save on time where I can. So you can eject the tip after each submersion as well. So yeah, I believe this is all that I can cover right now as of my rationale for programming the workflow as I did. And while SQWERTY is finishing off, it's going to take a couple of more minutes. I wanted to, wrap this experience up and also open the floor to any questions you might have, and this can direct our conversation further. Now, was automating PCR prep worth it for me? Is it worth it in general? I think it depends. It depends on what you value. Now, SQWERTY is not the fastest liquid handler on the market, but what it lacks in throughput, it makes up in cuteness and user-friendly and being accessible. Again, as a person who has zero experience with automated liquid handling before, I wouldn't even know how to approach one. I don't know coding. It all seems so complicated. And whenever I was looking at my colleagues, they would just need to call an engineer as soon as something goes wrong. This is why I was really happy to give SQWERTY a chance, because even I was able to get my head wrapped around it. Another thing that speaks about its ease of use is, as we were preparing SQWERTY to launch, we have given it to to one of our PhD students who had zero prior experience as well. And once we've sent SQWERTY to her, I was kind of booked my whole morning thinking I'll be answering questions on how to get it up and running. But what happened was, within 2 hours of getting SQWERTY out of the box, she was already running her first Bradford assay protocol. So yeah, it really speaks volumes about the accessibility we wanted to bring in, that we think the larger liquid handling systems lack. So whether it is the intuitiveness, setting up the workflows, how much time that takes versus how much the actual protocol takes, that might be something that differs from person to person. In terms of accuracy and repeatability, you know, there are studies published about the variability between people, even on different days, and different people in general. So I can imagine some, you know, poor undergrad and master students not always being consistent if it's a protocol that your lab needs to be repeating over and over and over again. By spending a little bit of time for a fraction of the price of larger liquid handling systems, you could get repeatability and consistency and traceability of all those protocols that you need to be doing on the daily. I believe I've covered all of the benefits that it put on me. I definitely don't mind waiting for SQWERTY to finish its thing while I'm sitting next to it and reading up on some papers. If it's you, you might be finally finishing off that manuscript or actually analyzing the data from the previous SQWERTY run. So not to keep you any longer, please, are there any questions? Can you all provide sample or make available some of these automated program protocols like the PCR ReadyMix, one that you have all have created? Very good question. We can absolutely provide some in advance. We are also really happy to work with you to find the settings that work for your reagents as well. We do think that it's best to try and test it yourself, because all of these different ready mixes, master mixes, coming from different suppliers can vary significantly in their properties and fluid dynamics. But definitely this is something that our application support can assist you with, including protocols as well. Hopefully that answers your question. Another question I've got there, have you all seen cross-contamination occur? Or what parameters do you recommend to eliminate cross-contamination? Another good question. So it does very much depend on how you program it. Again, it doesn't have the eyes or the rationale of a person, but if you use the interactive mode to program the insertion depth of your tube, how far the tip is going down, is it detecting the liquid? You can be eliminating any carryover and contaminating different wells by enabling the travel air gap. So it's a little bit of a complicated question, but the answer is we haven't seen it because we have used the parameters in order to kind of optimize it for our use. If you are concerned about it, you know, you can be ejecting tips more often, and you can be, controlling how you're optimizing and setting up the program. Hopefully that answers your question, but no, we haven't seen specific cross-contamination occur. And you can use the UV function. There was some water droplets on the tip after dispense. Is there a parameter that does a tip touch to the tube? Very good point. There is currently no tip touch. I do know what parameter you're referring to. I would give SQWERTY a little bit of a break because I have run the protocol design for all these reagents, but due to the health and safety concerns, we're working with food coloring right now. So while we were dispensing the water, some of the I've noticed that the fluid dynamics slowly gets bubbly. It is something that I would use different parameters like the blowout, increase the flow rate, and also increase where I'm aspirating and dispensing to kind of prevent that. So yeah, no tip touch yet, but there's other ways to mitigate it. How can you maintain accuracy if the range of volumes varies widely within the same workflow? Good question again. So you can think about it in different ways. Now, I'd first ask yourself, what is the protocol that you're trying to automate, and what are the critical steps of those workflows? So as I said, when it came to PCR Prep, I structured everything around the transfer of my template, which was so low volume that it kind of decided everything else for me. Now, if you're working with a broader range of volumes, it might be that you decide to do your overall really broad range 250 microliter module. But there's, yeah, definitely ways that you can mitigate it by thinking about how you want to be transferring your manual protocol to a robotic platform. You can also be using different volume tips, because they also do affect some volume of accuracy. This is all stuff that we would help you figure out when you're automating your first protocol. Another question, what are some of the other applications that SQWERTY is suitable for, and why? So, as I said, we are not in the throughput game. We are here for automating your menial, everyday, repetitive workflows. So your normalizations, inoculations, media preparations for multi-well plates, reagent addition. If you want that consistency, if you want to take some time back from having to pipette all day, definitely. I wish I could have used it during my undergrad when I had to pipette crystal violet all the time. Now I've got wrist injuries because of it. So, everything that is repetitive, manual, and you do on the daily, it's really worth it. We have also seen some different applications like MALDI-TOF target preparation, so the addition of different reagents before it goes to MALDI-TOF microbe ID, as well as Bradford assays, so protein concentrations, titrations, and so on. SQWERTY does have liquid level detection. Yes, apologies if I've omitted this. The benefits of liquid level detection is the precise drop-in of the tip exactly to where the surface is. It controls the aspiration much better, and it prevents you from setting the insertion depth manually. You can help it by kind of letting it know where roughly the surface of the liquid begins, to cut the amount of time it takes to get that pressure threshold. But yeah, it's to save you from having to figure out how far you need to be immersing, causing overflow like in a bath, and fluid displacement, sorry, and all the other things. So it's just ease of use and quality of life. What kind of technical support is available to help troubleshoot in workflows? Very good questions. So because we are new to this game, we are absolutely offering free application support to our SQWERTY users. So you'd be speaking to a small group of teams. We'd help troubleshoot any problems you might have with the instrument and your particular workflow. We would do, you know, the best that we can to get you up and running. But as I said, so far our experience has been that it's really user-friendly. You can wrap your head around it quite fast, and then everything else is, you know, little tips and tricks that you get as you're getting to know the instrument. So we're definitely here to help with any problems. Now, I don't know if we've got any more questions. Okay. If there's nothing else, I sincerely want to thank you for attending. Apologies for my... Oh no, actually, I planned all that abort run anyways. So hopefully I got you a flavour of what it's like to be using SQWERTY to automate your PCR prep. And if you've got any questions that you haven't managed to ask, or if you think about something later, please click on the screen when the pop-up comes to book a demo or book a chat with us. And yeah, we're looking forward to chatting with you. Thank you for joining and have a good rest of your day.